This presentation will talk about another type of an amine autacoid aside from histamine, the serotonin or the 5-hydroxytryptamine. So this presentation will focus on the pharmacology of serotonin as well as its associated antagonist. Presentation will talk about the history of serotonin, introduction to serotonin. We are also going to discuss about the sources of serotonin, the roles of serotonin, as well as its biosynthesis, distribution, and breakdown, the serotonin receptors. We are also going to discuss now about the pharmacological effects of the activation of these 5-HT receptors or the serotonin receptors, the agonist, and the serotonin antagonist. For the history of serotonin, in the year 1935, an Italian pharmacologist and chemist, Vittorio Erspamer, showed that an extract from the enterochromaffin cells of the gut made the intestines contract. 2 years later, Erspamer was able to show that it was a previously unknown amine, and he named that particular substance as enteramine. So this is Vittorio Erspamer at work in his laboratory at the University of Rome. In the year 1948, researchers Maurice M. Rapport, Arda Green, and Irvin Page of the Cleveland Clinic discovered a vasoconstrictor substance in the blood serum and named it serotonin. So these scientists, Rapport, Green, and Irvin Page, was credited to be the first researchers to isolate serotonin. So this diagram shows the early serotonin researchers. So among these are letter B, we have Arda Green. We have letter C, we have Maurice Rapport and Irvin Page. The hormone was chemically identified as 5-hydroxytryptamine, later by Rapport, and became known of more broadly as 5-HT. The word serotonin comes from combining the Latin word serum, meaning watery fluid, with the word tonic, which means medicine, and the suffix "in" or in, which is a common word-forming element in the science of chemistry that indicates an antibiotic, vitamin, hormone, or other substance. Serotonin was the name given to the vasoconstrictor substance which appeared in serum when blood clotted. Meanwhile, enteramine was the name given to the smooth muscle contracting substance present in the enterochromaffin cells of the gut mucosa. In the year 1950s, it was shown that enteramine was the same compound as serotonin, and they were chemically named as 5-hydroxytryptamine or 5-HT. The 5-HT or serotonin is formed and localized in 3 essential pools in the body. First, approximately 90% of the human and animal body's total serotonin is located in the enterochromaffin cells of the intestine. When we say enterochromaffin cell, that is a type of enteroendocrine and neuroendocrine cell, and it resides along the epithelium lining the lumen of the digestive tract, and it plays a crucial role in GIT regulation, particularly intestinal motility and secretion. So majority, 90% of the serotonin is found in these type of cells. Another is we also have a small number of neurons in the CNS and mast cells of rodents, along with histamine and heparin. So a small number or a small amount of serotonin can also be formed in these sites. We also have the blood platelets. So it is said that the blood platelets can sequester, they can store, and transport a remarkable concentration of serotonin. According to Cloutier et al. In 2018, the platelets do not synthesize serotonin, but they only utilize their serotonin transporter to capture circulatory serotonin. That is why they are capable of storing or sequestering the serotonin. In addition to the endogenous 5-HT reserve, the serotonin is also found in invertebrates in plants such as banana, pear, pineapple, tomato, and the stinging nettle. In the pineal gland, the 5-HT is converted to melatonin after acetylation and methylation. For the role or the functions of the endogenous serotonin, so this includes serotonin is a neurotransmitter in the CNS, particularly in the tryptaminergic nerves. Its deficiency causes depression and excess causes excitation. Serotonin is also a precursor molecule of the melatonin hormone. It also helps to regulate the tone and the motility of the GIT. The platelet serotonin serves as one of the mediators of blood clot formation. So while none of the following proposed functions are known for certain, the current evidence suggests that serotonin is involved in regulating gut motility, vomiting, migraine, appetite, body temperature, sleep, aggression, pain, mood, blood pressure, and endocrine function. In humans, serotonin is also known as the feel-good hormone in the gut, and it is also considered as the original happy hormone. For the synthesis of serotonin, so serotonin or 5-HT is synthesized from dietary L-tryptophan via a short metabolic pathway that involves two major enzymes. So the first enzyme that is involved in the synthesis of serotonin from L-tryptophan is the tryptophan hydroxylase. So tryptophan is hydroxylated by the enzyme tryptophan 5-hydroxylase to form 5-hydroxytryptophan or 5-HTP. The 5-HTP will then be decarboxylated by the enzyme 5-HTP decarboxylase to yield 5-HT or 5-hydroxytryptamine, serotonin, or 5-HT. So like catecholamines, 5-HT is also stored in storage granules and its uptake is also inhibited by reserpine. So when we say catecholamines, this refers to our epinephrine, norepinephrine, dopamine, and others. When we say reserpine, now this is an adrenergic blocking agent used to treat mild to moderate hypertension via the disruption of norepinephrine vesicular storage. Enzymes like monoamine oxidase, dehydrogenase, and aldehyde reductase help to metabolize 5-HT or serotonin. So we have here a diagram showing the synthesis and degradation of serotonin. So again, we have earlier said that serotonin, or the 5-HT, is derived from the dietary L-tryptophan by dehydroxylation and decarboxylation process. Later, the serotonin will be acted upon by the enzyme monoamine oxidase to yield 5-hydroxyindole acetaldehyde. The 5-hydroxyindole acetaldehyde will again be acted upon by the enzyme aldehyde dehydrogenase to yield the 5-hydroxyindole acetic acid, or the 5-HIAA. The serotonin can also be acted upon by another type of enzyme, the 5-HTN acetylase, to yield N-acetyl 5-HT. Further, it will be acted upon by another enzyme to yield melatonin, which is a hormone of the pineal gland. We also have here another schematic diagram of the biochemical pathway for the synthesis and degradation or catabolism of the serotonin 5-HT. So again, we have here the tryptophan. It will be acted upon by the enzyme hydroxylase to yield the 5-HTP, 5-hydroxytryptophan. And again, it will be acted upon by the enzyme decarboxylase to yield the serotonin, 5-hydroxytryptamine. The serotonin will be metabolized by the enzyme monoamine oxidase to yield 5-hydroxyindole acetaldehyde. And again, this will be acted upon by a dehydrogenase enzyme to yield 5-hydroxyindole acetic acid. Let's now proceed to the serotonergic or the 5-HT receptors. The present system of classifying the 5-HT receptors is based on molecular characterization and cloning of the receptors' complementary DNA. There are more receptors for serotonin than any other biogenic amine. At the present time, there are more than 14 receptor subtypes. Some of these have recently been discovered and their function is uncertain. The major categories of serotonin receptors include 5-HT1, 5-HT2, 5-HT3, and 5-HT4 to 7. All the 5-HT receptors except for 5-HT3 are G-protein coupled receptors, which function through decreasing or increasing the cyclic adenosine monophosphate or CAM production, or by generating inositol triphosphate IP3 or diacylglycerol DAG as second messengers. The 5-HT3 is a ligand-gated cation channel, which on activation elicits fast depolarization. The 5-HT1 receptor has 5 subtypes. This includes the 5-HT1A, 1B, 1C, 1D, and 1E. They are also known as autoreceptors. So when we say autoreceptors, this is a type of receptor located in the membranes of the presynaptic nerve cells. They inhibit the serotonergic neural activity in the brain. So this figure shows the 5-HT1A receptor. So the 5-HT1A receptor is a subtype of the serotonin receptor located in the presynaptic and the postsynaptic regions. So this figure shows a presynaptic 5-HT1A receptor. Again, the function of this, the activation of this type of receptor is It will induce neural inhibition and vasoconstriction. For the 5-HT2 receptor, so there are three subtypes, the 5-HT2A, 2B, and 2C. These are usually located in the CNS as well as in the peripheral sites, especially the vascular and the visceral smooth muscles, platelets, and the ANS neurons. The diagram shows a 3D model of the human 5-hydroxytryptamine receptor 2A. The effects of the activation of the 5-HT2 receptor are vasoconstriction, intestinal, bronchial, and uterine contraction, and platelet aggregation. The next serotonin receptor is the 5-HT3, and this has 5 subtypes. These are the 5-HT3A, 3B, 3C, 3D, and 3E. They are located in the CNS and in the peripheral nervous system. The activation of the 5-HT3 receptor induces emesis, gut peristalsis, bradycardia, transient hypotension, apnea, pain, and itching. Again, the 5-HT3 receptor is not a GPCR, but it is a ligand-gated cation channel. So aside from its location in the CNS and the PNS, it can also be located in extraneural locations like the lymphocytes, the monocytes, and the fetal tissue. We also have the 5-HT4 to 7 receptors. So for the 5-HT4 receptor, so it has no subtype, and they are located on the enteric nervous system, and it mediates intestinal secretion and augments peristalsis. For the 5-HT5, 6, and 7, so not much is known about these serotonergic receptors. Let's now proceed to the pharmacological effects of the activation of the 5-HT receptors. So let's start with the cardiovascular system. The arteries are constricted by direct action on the vascular smooth muscle as well as dilated through the EDRF release by the serotonin depending on the vascular bed and the basal tone. So the dilation due to the release of the EDRF. So when we say EDRF, it stands for the endothelium-derived relaxing factor. So in addition, the serotonin releases adrenaline from the adrenal medulla and evokes cardiovascular reflexes. So the net effect would be complex. The larger arteries and veins are characteristically constricted. In the microcirculation, serotonin dilates the arterioles and constricts the venules. You also have on the blood pressure. So there is a triphasic response. This is classically seen on rapid intravenous injection or infusion of serotonin in animals. So among the triphasic response include the early sharp fall in blood pressure. This is due to the coronary chemoreflex. After that, there is a brief rise in the blood pressure. This is due to the vasoconstriction and increased cardiac output. There is also a prolonged fall in the blood pressure. due to the arteriolar dilatation and extravasation of fluid. For the gastrointestinal tract, so serotonin increases motility of the small intestines and inhibits the motility of the stomach and the large intestines. For the respiratory tract, so there is a brief stimulation of respiration and hyperventilation. Large doses of serotonin can cause transient apnea and constriction of the bronchi. For the glands, serotonin inhibits gastric secretion, both acid and pepsin, but it increases mucus production. Hence, serotonin has an ulcer protective property. The next are on the nerve endings and the adrenal medulla. So the afferent nerve endings are activated by serotonin, causing tingling and pricking sensation and pain. Serotonin can also induce nausea and vomiting due to the depolarization of the visceral afferents. It is also less potent than histamine in releasing catecholamines from the adrenal medulla. For the platelets, by acting on the 5-HT2A receptors, serotonin causes changes in the shape of the platelets, but it is considered to be a weak aggregator. For the CNS, serotonin serves as a neurotransmitter. Primarily, it is inhibitory in nature. Direct injection in the brain produces sleepiness, changes in the body temperature, hunger, and a variety of behavioral effects. So it is said that the injected IV serotonin does not produce central effects because of poor entry across the blood-brain barrier. For the therapeutic uses, so serotonin has no therapeutic use. Let's now proceed to the serotonin agonist in nature. So these include the ergot alkaloids. So when we say ergot alkaloids, these include the ergotamine, ergonovine, and others, which occur in various combinations and are found in the fungi of the genus Claviceps, particularly the Claviceps purpurea. Another ergot is the ergovalin. This is produced by the fungus Acrimonium xenophyllum. For the pharmacological effects of the ergot alkaloids, so they can act both on the adrenergic and the serotonergic receptors, either as partial agonist and antagonist. They stimulate the smooth muscle of the gut, the uterus, the bronchi, and the blood vessels. For the therapeutic uses of the ergot alkaloids, so because of the complicated pharmacologic effects, ergot alkaloids with serotonin agonistic activity are in general not clinically useful. Ergonovine is used in obstetrics to stop postpartum hemorrhage and induce uterine involution. The next part would be the serotonin receptor modifying drugs. So these slides will feature some serotonin agonists and antagonists in veterinary medicine. So let's start with the GI prokinetic agents. So when we say GI prokinetic agents, these are drugs that increase the gastrointestinal tract motility by increasing acetylcholine release from the vagus nerve. So an example of a GI prokinetic agent is the cisapride. Cisapride is an agonist for the 5-HT4 receptor, and this is used for gastric intestinal stasis, reflux esophagitis, and constipation or megacolon in cats. We also have another GI prokinetic agent, the metoclopramide. Metoclopramide is a D2 or dopamine receptor antagonist and a 5-HT4 agonist. And this is used for treating vomiting disorders, reflux esophagitis, and gastric stasis or motility. Metoclopramide is an antiemetic agent and a dopamine D2 antagonist used in the treatment of gastroesophageal reflux disease, prevention of nausea and vomiting, and to stimulate gastric emptying. We also have the ciproheptadine. Ciproheptadine is a 5-serotonin or 5-HT2 antagonist and an H1 antihistamine. For its therapeutic uses, it is useful in cats as an appetite stimulant. It is also useful in the treatment of feline asthma or pruritus in cats. And it also has H1 antihistamine activity, and thus it is useful in managing hives. In horses, ciproheptadine is used for treating photic head shaking. You also have the ketanserine. Ketanserine is a 5-HT2 receptor antagonist. For its therapeutic uses, its potential use is to treat ergovalin-induced severe vasoconstriction. It also has a significant alpha-adrenergic blocking activity and thus reduces blood pressure. It can also be used to reduce the intraocular pressure in glaucoma. So other serotonin modifying drugs used are antidepressants.